Hi, everybody. Thank you for joining us today. I'm very pleased to have Ross Youngs, who is the founder and CEO of Biosortia. Ross and I spoke a few months ago, and what he is doing is really absolutely mind-blowing. I thought he would be a great guest for this podcast, and here he is. And Ross, I'm really excited for you to share your thinking. I mean, this is really, really big thinking, and it's interesting because on a previous episode, one of our entrepreneurs talked about, the you really do need to think big, and Ross is doing that here. And I think this is a really cool thing for him to share his vision and to, walk us through his ideas and potentially, whatever we can find an investor for him. So Ross, let me turn it over to you.
Well, thank you so much, and I really appreciate it. So, just a little bit about me before I jump in, just so there's a little bit of background. Environmental science, industrial engineering background, work in the medical imaging business, the video and optical disc business, really, and then I became a entrepreneur in 1988, hit the Inc. 500 5 years in a row with my first company, was named the nation's business person of the year by the Clinton administration
Wow.
in 1998, have won a ARPA-E, participated in DARPA, U.S. Air Force, NIH, and then we're supported also by Los Alamos National Lab. So a variety of experiences. I consider myself a generalist and more engineer and more processing engineer, which may be why this particular problem was looked at just a little bit differently
Yeah.
than those that are researchers that have a benchtop atmosphere to work from. I have an industrial scale atmosphere to work from. So when we really look at unlocking the microbiome, the dark matter, really not only for AI drug discovery, but the opportunity to directly access these molecules, we looked at it a totally different way. And that's our whole focus here, which is getting at what have been the most important drug-like molecules in cellular signaling. Microbes have that 4.2 billion years of evolutionary pre-optimization. And I'd like to point out that's a finite set of molecules, not an infinite set. In fact, we've only found 36,500 of these molecules. And using some references, some can say directly, indirectly, or inspirationally, that 36,500 turned into half the drugs on the shelf today.
Wow.
In one execution of our technology, which we'll tell you about in just a second, we expanded it from 36,500 to more than 110,000 novel small molecules from the microbiome. And we believe we're going to add millions as we execute over the next couple years. And as I explain what we're doing, why that is so relevant to finding next generation molecules to treat, cure disease. And you can expand it into agrochemical potentially, into sustainability, into cosmetics, into nutrition, into oil and gas. Every place life science plays, these molecules, these enzymes are critical for future biotechnology, and it could create an avalanche of new products because it has been looked at completely different.
Yeah, and I think that's what was so exciting to me when we first spoke.
So let's talk about the problem, because the problem really boils down to how well have we done. And this cost chart and this result chart down at the bottom right, it shows our costs have gone up 86% faster than inflation, basically since the 1970s, to find molecules to create drugs. We also we've seen a decline in new molecular entities. Now, this is with the world of computation, the world of synthetics, synthetic biology, synthetic chemistry. But the problem is all those indirect omics are looking at molecules that are possible, not the real molecules. So the golden age of pharmaceuticals was based on access to nature's best molecules, which we don't have right now.
Mm-hmm.
And that's what we're completely changing.
Mm-hmm.
Now, our solution is scale. Our solution is to go at the kind of quantity people thought would be impossible to obtain. But what we've learned from genomics is microbiomes exist all over this planet, and the metabolic pathways and the genes to create those molecules that exist in microbiomes overlap all over this planet. So even the human gut microbiome overlaps with the aquatic microbiome of the ocean by 73%.
Wow.
That is incredible to think. We know very little about the human microbiome and its molecules. If you go to the Human Metabolome database and you sort specifically for microbial molecules, it'll only list 144. That's it. So what we're looking at is we're looking at being able to uncover tens of thousands of those drug-like molecules circulating inside the human ecosystem, because they also circulate in other systems that are accessible. So when we talk about the kind of quantity, we're talking about, if you look at a 100 milliliter lab benchtop sample, that's a pretty good size sample. We're talking about 200 million times that typical sample. You can't go to a lab and say, we're going to process 8 Olympic-sized swimming pools of sample in order to pull out molecules that exist in sub-part per trillion, but at a quantity that still are biologically relevant to that microbiome. Therefore, this is why it's such a breakthrough. Capturing the microbiome without degradation, without lysing the cells, and with rapid preservation at such a scale that you have got this gold mine of opportunity. And I'll reference again, in one execution of our technology, working with Los Alamos National Lab, we found 73,500 novel molecules residing in one microbiome.
Wow.
So in the overall sense, this is kind of a layout of what we do. It's industrial microbiome mining, getting what best molecules nature has, putting it as the database, learning what that database is, and using AI. So we go to nature, we will do genomics, we will do preliminary chemistry. We want to find places that are unexplored but highly relevant for cellular signaling, specifically where microbes are abundant and where there is survival at stake. What kind of activities are these microbes doing to benefit their environment, to survive, to repair? Those are where we're going to find the most drug-like relevant molecules that impact the human.
Right.
Ultimately, I had mentioned getting it at the right quantity and quality. That is key. And you can see from this one photo, we're not talking about pulling up in a pickup truck and being able to process a microbiome. This was technology that took a decade to perfect and then ultimately peer review it. So obtaining those molecules is not using a 100-liter cup. Ultimately, processing it is key, and we'll talk a little bit about our black box technology. But even more important is if you have the analytics behind this and you have the molecules at hand, you can do things to those molecules to ultimately screen them and feed back more data into the engine. Right. And we'll talk about that right here. So basically what you saw where it says scout and find, the deconvolution is basically getting at this material's small molecules. So it's extractions, it's fractionations, it's analysis. From the analysis, we can build the kind of data sets that predict the smiles, predict derivatives. I'm going to skip de novo design right here, but predict its properties and its activities. Then we can prioritize, and we can either obtain these directly from the biomass, or if it's a more simple molecule that's been predicted, let's say a peptide, maybe a small molecule, easy to synthesize, we can make it. Then we can start to confirm the predictions,
Right.
get the structural elucidation confirmed, compare the predictions to the predictions we had. Then we get to model fit, prediction fit, and improve the models. This is a closed loop cycle based on nature's working molecules that ultimately allows us to do the de novo design
Wow.
because it's the best data set in the world for what really occurs at the cellular level. What's hugely advantaged here is where it says confirmed predictions.
Yep.
Our screening partners, which extend to people like Stanford, Ohio State, Michigan, Harvard, Case Western, George Mason. We've got universities all over the country that want our screening plates to run through their high throughput screening operations. That's all done on a confidential agreement. Obviously, if they find something, that gives us the opportunity to advance something within their ecosystem. But the data flows back in,
Right.
and that data will improve the predictions dramatically. And that's what's key, and that's what will generate a pipeline for customers. And that's really where we want to go, is we really want to find people who want to find next generation molecules.
Yep.
I would use the example of even things like metabolic sources. About 2 weeks ago, there was a report, and I think I'd posted it on LinkedIn, others had posted on LinkedIn, where they found a weight loss drug potential from a microbe, from a bacteria that looked very similar to Ozempic.
Right.
So nature's made a lot of these molecules. We've had no access to it.
Right.
And our approach right now, we're looking for that lead investor. If we put together that lead investor, we would be looking for money to move forward computationally and then move forward on the full scale execution. And I already talked a little bit about me, but I think this is a good place to pause because I have dozens of slides where we can talk about the details of this if you'd like to get into the details.
Sure. So Ross, I mean, AI has become, popularized over the last 18 months or so. Um, Prior to AI, would what you're doing have been possible, or would it be infinitely more difficult? Maybe go back to that slide, your flow chart, where you're almost like you're training your model and you're continuously refining, which I think is fascinating. Yeah, the
Yeah.
model improvements. Yeah, tell us a little bit about how AI is being deployed in this to
Yeah.
help you get the outcome that you're looking for.
It is so exciting knowing that they've been able to take these graphical interfaces, these graphical chips, and really turn these into the machines of AI. These large language models, whether you leverage large language models or you set up your own. We have a budget for one system that's about $120,000, and we believe we'll be able to populate that with nothing but real data. We don't have to use the synthetic data.
Yeah.
And that means the computational aspect of what we do is really not infinite,
Right.
which is a huge advantage. It's a finite data set that can be populated with exclusive data that will make this more predictive and more valuable. Now, we get to use a lot of the things that are open and available to enhance this. But you're right, this would have been harder, not impossible,
Yeah.
but the technologies that have really become available over the last several years make this very, and I'm not going to say easy, very doable in what we envision. And we've been able to do it non-automated easily. So as we automate this, which is our major goal, and then keep this data highly confidential, that will allow us to work with others that are looking for next generation solutions.
Yeah, yeah. And ultimately, you're discovering these new small molecules. That's the outcome, correct?
Well, that is absolutely. So when we go to this slide,
Yeah.
this is all about getting at nature's riches.
Yeah.
And I like to point out here a lot of people that have focused on omics, it's been great because it has provided us a massive amount of information needed as to where to go.
Yeah.
But genomics itself is the code.
Yeah.
And genomics itself does not necessarily lead directly to a result. Therefore, let's call it very convoluted from the code to the result. And most the code, you can't even predict a result.
Right.
It's still empty space.
Right.
In our particular case, we get to get the code, but we get to get the result.
Right.
We get the molecules that are existing within this microbiome. Ultimately, that's going to allow more connections to omics as well.
Right. Right. And these molecules, Ross, that you're discovering or you have the potential to discover, are they patentable? And can you lay any kind of claim to them?
Well, here's the beautiful part.
Yeah.
All of these have been unexplored by humans.
Yeah.
They haven't been used regionally or locally the way plants have. You don't have coverage because of some of the treaties that have occurred because these things are completely unknown. So finding a natural molecule doesn't guarantee a patent,
Yeah.
but understanding its activity allows patenting that natural molecule's activity.
Mm.
Derivatizing that natural molecule, which is not untypical because you start to build a portfolio around an activity.
Yep.
And we've seen this even naturally. One of our papers that got the Editor's Choice Award, Natural Products Editor's Choice Award, we found 3 unknown novel molecules in a lake in Ohio or lake in Michigan. And it ended up being very much like bufalin, except for it was cyanobufalin. The first time a microbe was ever identified as being a producer of bufalin. It's come from plants, it's come from animals. Likely it was horizontally transferred, who knows how many eons ago, into plants and animals. It became part of their repertoire of chemical production. But we found it within a microbe that had higher potency than we found sister molecules, natural sister molecules, then we could do derivatives of those. So you have the activity, you have the derivatives,
Yep.
and all of a sudden you then have what would be GMP. You start to build a real portfolio around IP that is related to a single or a range of activities.
Right. 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 Wow. And so who do you envision your customers being, Ross? What's your business model? What business do you want to be in?
Yeah, so we love the idea of being a platform.
Yeah.
And why we love that is we think we can be involved in solving problems that governments, academics, pharma companies, biotechs, agrochemical companies are all trying to solve. And we can work with defining a license, defining an area of focus. We don't want to be in a situation where we can't solve problems. We want to solve lots of them. So
Yeah.
we don't want to be the developer of these molecules. We want to be the source for building a starting IP that becomes defensible and becomes valuable. So we're only looking for what we would consider fair and typical upfronts, milestones, and royalties. And we are looking for upfronts from those that we've found an activity for and they decide to license it. But we're also looking for upfronts for those who want to carve out a segment and
Yeah.
use our engine and our capability to bring them molecules on that. And we certainly can see a lot of need for that because there are many untapped valuable spaces, or even if someone is in a space today and they want to, let's say, perfect it for the future, or make sure they have the next generation, or make sure they don't enhance a competitor, they may want to work with us on molecules of value, even if they already have a pipeline existing.
Yep. Yep. So basically, you're discovering molecules, and you can do it either on spec, right? Or you can do it sort of on a contracted basis. Would that sort of be an accurate depiction, Ross?
That is accurate. And
Yeah.
where we will get a lot of our initial discovery is from collaboration with academics. They have the high throughput screening capability. They have tested the heck out of synthetic molecules. What they haven't really had is the molecules from nature. And
Right.
us bringing those molecules of nature will probably uncover a ridiculous amount of unknown activity, unknown molecules, probably novel structures, expansion of the chemical classes. We just think that this is uh, going to play across all discovery segments. And we want to ensure that it does that.
So I'm just curious, Ross, tell us about the equipment in the upper right-hand corner. The trailer in the, is that a pump or tell us a little bit about that?
Yeah, so when you talk about moving a natural microbiome at scale, and this is an aquatic setup, but when you think about being able to have a microbiome at scale, liquid is the transport, liquid is what is required around every microbe generally, or microbiome. So in this particular case, we're going to sources that already exist. And the nice part about that is we can pull 8 Olympic-sized swimming pools out, and we will have 1,000 grams dry weight of a microbiome. It's a starting quantity that is just insane. That's why we need that industrial scale
Right.
to process it. So
Right.
we get it at industrial scale. We'll be processing at industrial scale. And the whole key thing here is not to be the research group,
Right.
to be the executioner of getting and making it available to the researchers. Now, is there the opportunity that Biosortia, in a fairly short period of time, working with so many academics, we could be something like a venture studio?
Yeah.
Because if Stanford ends up picking up a molecule ours and they start to move that forward and that becomes a NewCo, we could be in a position where we could take an interest in that NewCo, but we don't want to be that developer.
Yeah.
So we may support the academic side of this. When you look at this equipment, we're processing 1 to 2 million liters a day.
Wow.
Now, there's not a lot of systems that do that. Water and wastewater systems do that, but the problem is they will lyse the cells and they will have too long of a dwell time. Almost everyone that's looked at natural microbiomes before have looked at sludge.
Yeah.
Microbes have about a 15-minute change everything type of time frame.
Hmm.
If you take it out of its existing habitat where it's stable, where it's metabolically stable,
Yeah.
and you do something to it, it is reacting quickly. That whole microbiome's reacting quickly. And people that have looked at sludge see sludge. That's the
Right.
decomposition. And that is a very different kind of microbiome than a living, breathing microbiome. So it is so critical to
Gotcha.
obtain a living, breathing microbiome and putting it in suspension immediately.
Yeah. So is that equipment proprietary to you, Ross? Did you invent it?
Yeah, it's proprietary. We ended up on our Department of Energy grant,
Yeah.
which was the first attempt to collect microbes at scale. That originally was about doing it at very low energy. Now, what we learned is, and we did receive a $6.3 million grant that helped us really move this technology forward, but that was generation one. What you're seeing there is generation four technology.
Gotcha.
But generation one technology, which was the last one we disclosed and the ones we patented, we've received 4 patents on that. That particular technology had the equivalent of removing a ton of microbiome from an aquatic source, not at what was using centrifuges, which was about $3,500 per ton, but using this low energy approach, which worked out to $1.92 per ton.
Oh, wow.
So we had a significant reduction in the cost of removing microbes from so much water. Now, That particular technology was focused on biofuels, bioproducts. We were working at the time with a researcher at University of South Carolina, Charleston, and the Medical University of Charleston, I guess I should say. And they took some of our biomass and started to explore it. And what we found is having a low energy recovery with a rapid suspension of their metabolic activity allowed that researcher to basically say, oh my God, I've never seen so many novel molecules in my life. And that really started us down this pathway. Is it for the first time said this hidden information in nature is actually available?
Yeah. Wow. So tell us a little bit about what does the next 6 to 12 months look like for you, Ross? If and when you get your funding, what are your plans?
Yeah, so the
Little,
next 6 months to us is we were worried we were at a critical time, especially after the start of COVID. We thought we were going to get completely funded. We had some amazing engagement right before COVID,
yep,
and we've been on pause since.
yep,
Pause means we've got the technology proven and scaled to get microbiomes. What we've learned during that pause period is we can now move forward computationally.
yep,
We would like to move forward computationally today,
yep,
but that requires us engaging with the right people. So I've put a substantial amount of my own personal wealth
yep,
into this business by writing checks in excess of
yep,
$5 million to keep us moving forward. We have the resources to be on pause indefinitely,
yep.
but executing computationally, I think will show anyone and everyone that the promise of this technology is real. Executing the full-scale microbiome mining facility means we're putting together a series of extraction technologies
Mm,
with basically every downstream technology to fractionate, to well plate, and then to screen. Now we'll do initial preliminary screening internally, but ultimately we want to leverage our partners, and that could include biotechs,
yep.
pharmas, and academia on our screening of our molecules themselves to feed back in the data so that it becomes that circular continuous improvement.
Right. And needless to say, the upside has to be enormous, correct?
We believe it is. I would say this with the lull in access to nature's most valuable cellular signaling molecules, that if we have a landslide of
Yep.
access to these molecules, we will see a proportionate increase in new IP that could impact a variety of these life science areas. So we've made the bold claim that within 10 years of industrial scale microbiome mining, access to this dark matter, that most new products will come from this technology, which is a incredibly bold statement considering the hundreds of billions of dollars that have gone into indirect omics.
Sure.
But when we look at what these molecules can do, and these aren't without issues.
Yep.
These molecules need to be delivered to the right place, which there's technologies advancing for that. A lot of times you have fragile molecules, and you've got to be able to take into advantage of that. Some of these are complex, so you've got to be able to have a GMP to obtain it. So
Yep.
these are not completely a panacea that is no barriers in the way, but this is an opening of the reality that nature's secrets are really going to unfold to us, and it will be leveraged across all the omics, but it'll also be leveraged with the direct pathway to the chemistry.
Yeah, yeah. So Ross, one final question for you, because I think, what really impressed me about talking to you the first time was just sort of the vision and the vastness of it, such a big vision. And it's, again, it's not a simple one. So what advice would you have for other entrepreneurs, scientists, engineers that have big visions that are trying to bring something really scientific to market that they believe can be really a changer for mankind, if you will.
Yeah, if I had to say anything where I think I could have a impact on research and research organizations, we build specialists,
Yeah.
and specialists have to become more specialized in order to carry weight within their industry. So, and I love that specialization, but we rely dramatically on whether it's just the ego or the expertise of the leadership potential within a research organization to say this is what we're going to do.
Yeah.
And it's harder to create multifunctional teams.
Yeah.
And I am an absolute believer in listening to what others have to say from their form of expertise related to a topic they know nothing about,
Mm-hmm.
rather than dismiss them as you know nothing about this.
Right.
And I think that when you look at a Harvard Business Review and it talked about innovation in general, 50% does come from the specialist. The other 50% comes from the generalist.
Right.
And our history in life sciences suck.
Right.
Any other industry in the world would have been collapsed and shut down based on cost and performance.
Right. Right.
It has done a terrible job of bringing products to market.
Right.
And because of that, they reuse products.
Right.
They modify generally. And I think that there is the opportunity to bring lots of new products to the market. And it's not doing the same thing.
Mm-hmm.
It's opening up and trying to drive research with more exploratory and more minds involved that are not completely focused on one issue. I like the reality that almost every major breakthrough in life sciences came from the edge or outside of thought leadership, or came completely from outside of life sciences. And that's hard to believe, but that's really true. The breakthroughs are not coming from the centrist, well thought out positions. They're coming from the edges or outside the edges.
Fascinating. Well, Ross, thanks so much for sharing your story. I'll be sure to get it out to our community, and I wish you all the best of success going forward.
I appreciate that greatly. And I think the work you're doing, I've seen so many of your podcasts and some of the work you've written. I really appreciate you being out there and giving opportunity for messages to be heard. So thank you.
Thanks, Ross. Take care.